Hi everybody, let's get started with the cardiovascular system, in particular the blood segment of it. Now, blood is the perfect link between the respiratory system and the cardiovascular system. We've already talked a bit about hemoglobin and how we carry oxygen and carbon dioxide and gas exchange, and blood is certainly the biggest part of that. So let's first start with the function of blood, and then we'll get into blood components. The function of blood includes transport. So it delivers oxygen from the lungs and nutrients from the digestive tract to all body cells. We've talked about that with the digestive system. It transports metabolic waste products from cells to elimination sites like the lungs to breathe out CO2 and to the kidneys to get rid of waste in urine. It also transports hormones from the endocrine system, which we've talked about, to their target organs. So lots of transport happening. We also have a lot of protection that occurs. The protective functions of blood include inflammation, which certainly is part of our immune system as well. It helps to prevent infection, and it's the white blood cells, antibodies, and other blood proteins that help with this. We also don't bleed to death because there's platelets within our blood that helps with homeostasis. And then we have regulatory functions of blood, like fluid distribution. And it makes sure that we have an adequate fluid volume in our circulatory system. Like, we want to make sure you don't have too much blood in your feet and enough blood in your brain. The blood we've already mentioned when we talked about the respiratory system, that we do have acid and base buffering within it. And that not only is for the blood itself, but it helps to buffer all sorts of places, our tissues and our organs. So let's talk about the components of blood, and the first of those is plasma. Now, plasma is clear, kind of a light yellow color. It's the extracellular matrix of blood that contains at least 90% water. It has proteins and nutrients, electrolytes, waste, hormones, and gases. Everything that the blood's carrying around is within that plasma. And these are the tiny components that are dissolved in it. We also have formed elements, and these are the ones that are visible using a microscope. They're not dissolved in the plasma, but they are carried in the plasma. So the formed elements are suspended in the plasma, and plasma is a non-living fluid matrix. If we spin down a sample of blood in this centrifuge, you can see from this image here down below what we would end up with. So in the lab, we can do this, or in a hospital, they spin down the blood. It's a very quick process, spin it down for just a couple minutes. And what you'll find is that the centrifuge packs down the heavier formed elements, and the less dense plasma stays at the top. So most of the reddish mass at the bottom, you can see, are the erythrocytes, or the red blood cells that transport oxygen for us. They're the ones that contain hemoglobin. So that's this down here. Then we have this little tiny layer called the buffy coat, and the buffy coat is present at the erythrocyte plasma junction. This layer contains all of our white blood cells, or our leukocytes, and also all of our platelets. So it doesn't look like very much, but truly they do a lot of good things for us. Then we have plasma, which is at the top, and again, this is mostly water, 92%, depends on the person, and with proteins and other solutes dissolved within it. Now, normal hematocrit, which is the number or the percentage of red blood cells, erythrocytes, in our blood, when you spin it down, normal hematocrit values are somewhere around 47% for men, plus or minus 5%, and for women, it's about 42%, plus or minus 5%. Leukocytes, or white blood cells, and platelets are less than 1% of the whole sample, and plasma is somewhere around 55% or so. Now, in Colorado, I already mentioned when we talked about the respiratory system, at higher elevations like us, high altitude, we do have a slightly higher percentage of red blood cells, or a higher hematocrit. So we might have something like 50% plasma rather than 55%. It really just depends on the person, your age, your gender, how much iron you have in your diet, all sorts of things. So let's talk about plasma first. Again, it's kind of a light yellow or straw colored fluid, mostly water, but it does contain over 100 different dissolved solutes, like those nutrients, gases, hormone, wastes, all electrolytes, all of the things I've already mentioned. Let's talk primarily about how it has some very specific plasma proteins, and we'll just mention the most abundant ones. So the most abundant one is albumin. And albumin, if you look on this table over here, here's albumin. It's about 60% of all of the plasma proteins, and it does a number of different things, including acting as a carrier to shuttle certain molecules through the blood and circulation. It's the major blood protein that contributes to plasma osmotic pressure, which is the pressure that helps to keep water in your bloodstream. The composition of plasma varies continuously as cells remove or add substances in the blood. So we have other different plasma proteins, including globulins. This is about 36% or so, maybe a little more. These are made by the liver. You know I love the liver, so I have to give props to the liver. Most of these are transport proteins that bind to lipids, fat-soluble vitamins, and things like that. But we also have gamma globulins, which are antibodies, which we'll talk about later when we get to the immune system. We also have fibrinogen present in our plasma protein, and fibrinogen is about 4% of our plasma protein. Fibrinogen is produced by the liver as well, go liver, and it forms threads in a blood clot, which we're going to get to here in just a few minutes. So we also have lots of nutrients and electrolytes, respiratory gases that are all present in plasma, as well as enzymes and hormones that are on the move, being carried to where they need to go to their target tissues and cells. So let's talk about the formed elements, and we will start with erythrocytes. Now, it has cyte in the name, but it is technically not a cell. And that's because red blood cells, or erythrocytes, don't have a nucleus. Instead, they have this sort of depressed center, which is where the nucleus once was when it was first formed. And as part of the production of red blood cells, the red blood cell, or the erythrocyte, actually loses its nucleus. Now that seems strange, but the erythrocyte does have a lot of surface area, and really it is just a big bag of hemoglobin. It's about 97% hemoglobin, and then the rest is maybe plasma membrane and things like that. But really, there are no organelles and no nucleus in a red blood cell. It truly is just a transporter for oxygen and carbon dioxide. And if they don't have any organelles or a nucleus of their own, they don't consume any oxygen, and that makes them very efficient transporters. That means they don't consume anything, they just carry that oxygen and CO2 where it needs to go, and they don't participate in that at all. So, the hematocrit, like I said, is the percentage of red blood cells that are present in your bloodstream, and it's somewhere between 38 to 56%, with us at high altitude erring on the high side of that percentage. Now again, women are slightly lower, men are slightly higher, and part of that has to do with the fact that women do menstruate. And so we have a slightly lower percentage of erythrocytes, certainly at particular times of the month. Now, the erythrocyte does have some other things to talk about. I wanted to mention blood doping because it's been such a big deal in the past decade or so with athletes. Blood doping is basically when an athlete will train at high altitude, take out a pint or two of blood, spin it down and save the erythrocytes until they're about ready to complete a race or an event or whatever their sport is. Right before they are about to compete, they'll then put those erythrocytes, those concentrated erythrocytes, back into their bloodstream. And this makes it so that they can deliver an incredible amount of oxygen to their skeletal muscles, and they have a high performance. It is really dangerous, though, if you think about it. It does make blood goopy. In Colorado, we do have more viscous or goopy blood. It makes us slightly higher at risk of strokes and things like that. But especially for athletes that are blood doping, making their blood so viscous with all of those erythrocytes, it's incredibly dangerous. It's also illegal. So I will mention Lance Armstrong. He was found guilty of blood doping, but it was not that particular kind of blood doping. Instead of this, which is illegal, he was actually having the hormone erythropoietin, or EPO. He was injecting that into his bloodstream, which is a hormone that our kidneys make normally that tells our bone marrow to make more erythrocytes. So he was using a natural hormone to stimulate his own bone marrow to make red blood cells. And the way that he got caught, if I remember correctly, is somebody actually saw the vials of EPO in his refrigerator. Now, red blood cells are very tiny. They're about 7.5 micrometers. And sometimes when they go through the tiniest capillaries, they have to line up single file. And this name is a rouleau, lining up single file to go through the teeniest, tiniest little capillaries. Now, when your blood is oxygenated, that is, your erythrocytes are loaded with the maximum amount of oxygen, your blood is a dark red. And this is obvious when you bleed, right? The atmosphere exposes all of the hemoglobin in your red blood cells to oxygen, and it is that dark red color. Now, deoxygenated means that some oxygen has been lost and some CO2 has been gained, and a lot of people think that this blood is blue, but it is not. It is still a dark red. The reason it looks blue is because you're seeing veins through lots of different tissues and things like that in your arms and your legs. It's a slightly different shade of red, but it certainly isn't blue. How do we know that? Well, you could put some blood in a container and suck out all the oxygen. It would be very obvious very quickly that you are not blue-blooded, unless maybe you're a Smurf. And if you are, send me an email. I would like to talk to you. All right, so hematopoiesis is the process by which we make blood cells. And all blood cells start from a common blood stem cell called a hemocytoblast. All formed elements come from this. So this is a blood stem cell. It is found in your bone marrow, your red bone marrow specifically, if you remember your anatomy and physiology 1. It is a stem cell for all formed elements. Now, erythropoiesis specifically is the process by which we make erythrocytes, or red blood cells. And you can see this on the left side of this chart. Now, I'm not going to require that you memorize the progenitor cell and the proerythroblasts and all that stuff, but I do want you to know some of the main players, what happens during erythropoiesis. And I want you to know specifically that EPO, or erythropoietin, is the hormone released by the kidneys that tells us to make red blood cells, or erythrocytes. Now, the stimulus for this to make EPO, first and foremost, is that we always make a small amount of EPO. It's constantly circulating in our blood at all times and sustains red blood cell production at just a basal rate. You have to have red blood cells. They have to turn over. You have to make new ones all the time. The kidneys play the major role in EPO production, although I gotta give props to the liver. It makes a little bit too. All right, so there are things that cause more EPO to be created, and one of those is when certain kidney cells become hypoxic or oxygen deficient. Oxygen-sensitive enzymes are unable to carry out their normal functions, and this causes more EPO to be created and released. So a drop in normal blood oxygen levels triggers EPO formation. And this could be from reduced red blood cell numbers due to hemorrhaging, like bleeding, or excessive red blood cell destruction. It could be due to insufficient amounts of hemoglobin per red blood cell, something like an iron deficiency. Or it could just be from reduced availability of oxygen, which can occur like it does here in Colorado at high altitudes, or maybe if you have something like pneumonia or some type of lung infection. Now, on the opposite side of the spectrum, too many erythrocytes or too much oxygen in the bloodstream depresses EPO production. So let's, one last thing I want you to see on this left-hand side is that at the last part of erythrocyte formation is this ejection of the nucleus and the organelles die. So we have what looks like this normal blast spitting its guts out. It's getting rid of the nucleus so that all of the erythrocytes can be very efficient oxygen and CO2 transporters. The life cycle of a normal red blood cell is about somewhere around 90 days or so it really depends upon the person, but we do have to constantly make more. 90 days, 120 days, it depends on which book you look in. But low oxygen levels, let's start at the very beginning. Low oxygen levels in the blood stimulate your kidneys to produce EPO, or erythropoietin. The EPO levels rise in the blood, and then the red bone marrow starts making more red blood cells, erythropoiesis. As part of the last part of the process, remember, the erythrocytes get rid of the normal blasts, get rid of the nucleus, and we have new erythrocytes enter the bloodstream, and they stay there somewhere 90 to 120 days. Eventually, they get beat up and damaged and old, and they have to be taken care of, taken out of distribution, recycled. And you know who does most of this? Of course, it's the liver, right? The liver is the primary recycler and destroyer of dying or aged erythrocytes. So a lot of things that the liver does has to do with recycling and reusing things. So within the liver, we have macrophages that engulf and destroy these erythrocytes. And then the heme of their hemoglobin is split off from the globin. The iron is taken, bound to a protein, and stored to be reused. And then the rest of the heme group is degraded to something called bilirubin, which is a yellow pigment, if you remember, that's a precursor to bile. Liver cells will take up that bilirubin and turn it into bile, right? And then we use that to digest fats. So the protein or globin part of hemoglobin is metabolized and broken down into amino acids, sent back into circulation, and we can even use that to make new erythrocytes or red blood cells. It really is amazing how well we recycle. So 90 to 120 days is pretty much the average lifespan of a red blood cell or an erythrocyte. So it's important that we have constant levels of EPO to keep new ones being made all the time. So here's the white blood cells. There are 5 different types of white blood cells: the granulocytes and the agranulocytes. Granulocytes contain obvious membrane-bound cytoplasmic granules, and agranulocytes lack obvious ones. Now they all have a little bit, but you can see them much more obviously in our granulocytes. Now the granulocytes include neutrophils, eosinophils, and basophils. All are pretty round in shape, and they are larger and don't live nearly as long as red blood cells. They usually have nuclei that are in lobes, or these little sort of rounded bunchy pieces, compartments of the nucleus. And we identify all of our white blood cells based upon the nucleus and the color of the white blood cell itself. So the order of the most abundant white blood cell to the least abundant are neutrophils, lymphocytes, monocytes, eosinophils, basophils, in that order, with neutrophils being the most abundant, somewhere between 60 to 70% of all white blood cells, and then the most rare ones are the basophils, 0.5 to 1% total. A way to remember this order is a simple little mnemonic: never let monkeys eat bananas, or NLMEB. That's the order of most abundant to least abundant white blood cells. So let's start with neutrophils, the first of our granulocytes. Neutrophil cytoplasm contains very fine granules that are a little bit hard to see, but they are there. Neutrophils, like I said, are the most abundant of all of the white blood cells. Now, all of the granules together give the cytoplasm sort of a lilac color or a light purple color, and some of these granules contain hydrolytic enzymes like lysosomes. There are also some smaller granules that have antimicrobial proteins, those defensins that I've talked about previously. Now, neutrophils usually have somewhere between 3 to 6 lobes. The most we've ever found in lab is 6. I'm telling you, someday I'm going to find a 7. It's going to be a happy day, but usually 3 to 6 lobes. They're usually called polymorphonuclear leukocytes, or PMNs. Sometimes they just call them polys because their nucleus is so varied depending on which one it is. So neutrophils are our body's bacteria slayers, and their numbers increase exponentially during bacterial infections, everything from meningitis to appendicitis. Neutrophils are chemically attracted to inflammation sites, and they are active phagocytes, which we're going to talk about more when we get to the immune system. They are particularly good at ingesting bacteria and some fungi, and they use a vesicle called a phagosome to do this. We're also going to talk about, when we get to the immune system, how a way that neutrophils kill bacteria is a respiratory burst, which basically the cells, the neutrophils themselves, they metabolize oxygen and they produce this germ killer oxidizing substance like bleach and hydrogen peroxide mixed together. And they literally just spray or shoot it at the whatever it is they're trying to destroy. So we'll get back to that more during the immune system. Now, we're going to talk about eosinophils, which are also part of the immune system. Eosinophils are about 2 to 4% of all white blood cells, about the same size as a neutrophil. Their nucleus usually has 2 lobes that are connected in the middle by this little band in between. And there are large, coarse granules that stain sort of an orangey red color. These are lysosomes, and they have a lot of different digestive enzymes, but they're not designed to digest bacteria. Instead, eosinophils are really designed to attack and destroy parasitic worms like flatworms and roundworms, things like pinworms and hookworms, nasty, that are just too large to be pulled in and phagocytosed. The worms are ingested in food, like if you eat a lot of raw fish or sushi or something. Sometimes they come in through the skin. But eosinophils in particular live and reside in loose connective tissue in these same body sites. And when they encounter a parasitic worm, they sort of cluster around it and release the enzymes from their granules onto the parasite surface, and it dissolves those nasty worms. So there are other roles that eosinophils have, usually involving allergies and asthma, and we'll be talking about those more later when we get to the immune system. Basophils are the rarest of all the white blood cells, again 0.5 to 1% of the entire population, and their cytoplasm stains really, really dark, so dark that the whole thing almost looks completely dark purple. They have these histamine-containing granules that stain really dark, and histamine is an inflammatory chemical that not only is a vasodilator that makes blood vessels dilate or open, but it also attracts other white blood cells to sites of inflammation. If you think about it, antihistamine is a drug that counters this effect. So the nucleus is in there. It's sort of U-shaped, but it's hard to see because of how darkly the little granules stain. So granulated cells similar to basophils called mast cells are also found in connective tissue. These act a lot like basophils, but one of the main things that they do is they cause cells to release histamine. They'll cause basophils to release histamine. All right. So now let's talk about our agranulocytes. These include lymphocytes and monocytes, and they don't have really hugely visible cytoplasmic granules. Their nuclei are usually either a sphere or a kidney shaped. So lymphocytes are about 20 to 25% of all of our white blood cells, and there's 2 different types, B and T lymphocytes. And for our purposes, you can't really tell the difference just by looking. But these are huge players in our immune system, coordinating immune cell activity. B lymphocytes make antibodies. T lymphocytes attack pathogens and infected cells. I mean, these really are the key players in our immune system, which we will get to know and love as soon as we get there. Monocytes are about 3 to 8% of our total white blood cells. And while the lymphocyte has a perfectly spherical nucleus, look at the monocyte with its kidney-shaped one. It looks very cute, like a little C or a kidney-shaped nucleus. Monocytes are macrophages. These really, when they go to whatever organ or place in the body they need to be, they are then called macrophages, which their job is to phagocytose infected cells, bacteria, parasites, all sorts of things. And again, we will talk about these more as we go along. We've already mentioned macrophages in the respiratory system that are in the alveoli themselves, and they were once monocytes. So we're back to hematopoiesis, and we're going to focus specifically on leukopoiesis, the production of white blood cells. And this is stimulated by chemical messengers. Now these chemical messengers, which can either be paracrines or hormones, they are either interleukins or colony stimulating factors, or CSFs. Interleukins are numbered. They all have names, IL-3, IL-5. And then the CSFs are named for the leukocyte population that they stimulate. Something like granulocyte CSF stimulates productions of granulocytes. So what I want you to see here at the top, we have the hemocytoblast that everything comes from. We have the myeloid stem cell line, which results in the production of erythrocytes. Also, the bulk of our white blood cells, all of them except for the T lymphocytes. But the T lymphocytes have their own lymphoid stem cell. And that's important because these are such adaptive, specific killers when it comes to our immune system. So, interleukins and those CSFs, or colony stimulating factors, are released by supporting cells in bone marrow and by mature white blood cells themselves. It makes sense that interleukin, what that means is in between white blood cells. So again, I don't need you to memorize every single step, but I do need you to know the stem cell lines that these structures come from, as well as the categories of hormones or chemical messengers that play a role in regulating this. Platelets are our last type of formed element, and these are small fragments of bone marrow cells called megakaryocytes. Again, a platelet is not a cell. Instead, it is little tiny pinchy pieces of the plasma membrane of these large megakaryocytes. Thrombopoiesis is the actual production of these platelets, and you can see here we have thrombopoiesis. And our megakaryocyte here, little pinchy pieces of it, are the last step in thrombopoiesis, creating those tiny little platelets. So platelets are not cells. The major hormone that produces or stimulates the production of thrombocytes, which again are not cells, is thrombopoietin. What's really interesting about the production of platelets is that we do make this giant megakaryocyte, and it presses against a specialized type of capillary that we have in our red bone marrow that has lots of huge holes in it called sinusoids. It sends out little cytoplasmic extensions through the capillary holes, those sinusoids, into the bloodstream. And these extensions, instead of being little pinches, they actually rupture and they release the platelets like leaves blowing off of a tree. They seed the blood with platelets, and then the plasma membrane quickly seals around the cytoplasm, just seals back up again. It's really interesting. So we're going to talk about hemostasis, right? This is what happens if a blood vessel wall breaks. We need to have a whole series of reactions to accomplish the stopping of the bleeding, which is specifically what hemostasis means. If we didn't stop the bleeding or plug the hole, we would quickly bleed out our entire blood volume, even from the smallest of cuts. Now, speaking of blood volume, we should talk about how much blood we actually have in our body. Again, blood is the only fluid tissue in the body, and we have about 8% of our body weight is blood. For women, it's about 4 to 5 liters, and men, it's about 5 to 6 liters. I guess if we can just agree somewhere on average about 5 liters, that's how much blood you have in your whole body. Your blood temperature inside of your body is 100.4 degrees Fahrenheit or 38 degrees Celsius. Now, it does read a little bit lower when you take your temperature from outside of your body, but generally we don't stab you with the thermometer to get a core body temperature of your blood. But if you did, it would be somewhere about 100.4 degrees Fahrenheit or 38 degrees Celsius. Now, don't forget, blood is a connective tissue. You should have learned that in A&P1. So let's talk about the process of hemostasis, or preventing the loss of all 5 liters of our blood. Now, the first step is a vascular spasm. This is where the damaged blood vessels respond to injury by a very quick constriction called a vasoconstriction. This lasts somewhere around 20 or 30 minutes, allowing time for the next 2 steps to occur. Now, this vascular spasm, the trigger is direct injury to the vascular smooth muscle, that is the smooth muscle that's in the vessels themselves, but also by chemicals released by damaged endothelial cells, which are the lining of the capillaries or the blood vessels. Also by activated platelets, which we'll talk about next, and reflexes initiated by pain receptors in the area. The spasm mechanism is more and more efficient as the amount of tissue damage increases. And it's obviously most effective in smaller blood vessels. If somebody cuts your carotid artery, there's not any sort of constriction in this little vascular spasm that's probably going to save your life. But in a small, strongly constricted artery, this can significantly reduce blood loss for that first 20 to 30 minutes. And step 2 is the platelet plug formation. So in this second step of platelet plug formation, platelets play a key role in hemostasis by aggregating or sticking together, and they form a plug that temporarily seals the break in the blood vessel wall so that you don't bleed out. So as a rule, platelets just flowing through your bloodstream don't stick together, and they don't stick to the lining of blood vessels. That's called endothelium. So the way that this works is that intact blood vessels, their lining, those endothelial cells, they constantly release nitric oxide and a hormone called prostacyclin. Both of these chemicals together prevent platelets from clumping or aggregating together. And this is in undamaged tissue. But when tissue gets damaged, when the blood vessels get damaged, that endothelium that lines our vessels and in the collagen fibers of the underlying connective tissue are exposed, platelets will adhere to the collagen fibers. And this is because a large plasma protein called von Willebrand factor stabilizes those bound platelets and forms a bridge between the collagen and the platelets. And when this happens, the platelets become activated. They swell, they get big spikes on them, and they become really sticky. In addition, they start to release chemical messengers called ADP, or adenosine diphosphate, and serotonin and thromboxane A2. So adenosine diphosphate is an aggregating agent that causes more platelets to stick to the area and release their contents. And serotonin and thromboxane A2, they're little messengers that enhance vascular spasm and also the platelet aggregation. So as more platelets aggregate, they release more chemicals, aggregating more platelets, and so on. And this is what we call a positive feedback mechanism, if you remember that from your general biology and A&P 1 courses. And within about 1 minute, a platelet plug gets built up that helps and plugs the area that further reduces blood loss. Now the next step, step 3, is coagulation. And I admit I'm not going to go through every single mini step of this. You're welcome. But if you want to watch some videos about hemostasis, more than likely they will go through the 2 pathways involved that use clotting factors. So let me just give you a general overview of it, but you're not expected to memorize all of the different clotting factors and in which order they come together. So coagulation is a multi-step process that uses clotting factors. They're numbered 1 through 13, Roman numerals. Most of them are made in the liver, and there's a whole cascade that happens, which I'm not going to go into, to form this particular molecule called prothrombin activator. And this prothrombin activator, let me circle it here. When it gets formed, it converts a protein called prothrombin to thrombin. Now, prothrombin was a plasma protein that was just floating around, but when this prothrombin activator catalyzes prothrombin into thrombin, thrombin then catalyzes fibrinogen to fibrin. Now, fibrinogen are just little molecules that are already in your blood plasma as well. But when this whole process occurs, we now have thrombin catalyze the joining of fibrinogen molecules into fibrin, and that creates a mesh. Fibrin is a polymer. This fibrin mesh traps blood cells and seals the hole even more. And if this wasn't enough, this fibrin mesh has something called a clot retraction and repair. There are contractile proteins within the clot that sort of cinch up. They have contractile proteins, just like we saw in muscle, actin and myosin, and they contract much in the same manner as smooth muscle cells. So as the platelets contract, they pull on the surrounding fibrin strands, squeezing the serum and sort of squeezing out the serum, and they make that clot just really compact, drawing the ruptured edges of the blood vessel even more closely together. As this clot retraction is happening, the vessel is also healing. This stuff called PDGF, or platelet-derived growth factor, is released by platelets, and it stimulates the smooth muscle cells in the surrounding vessels and the fibroblasts to divide and rebuild that vessel wall or vessel walls that were damaged. So as the fibroblasts form connective tissue patch in the injured area, endothelial cells, which are stimulated by a vascular endothelial growth factor, they multiply and restore the lining of the blood vessels that were damaged. All right, the next step is fibrinolysis. So a clot is not permanent. We have to actually get rid of it. Fibrinolysis removes unneeded clots when healing is done. So this is important. We don't want to just have old clots lying around. We have to have fibrinolysis, where we allow those blood vessels to flow again and become completely unblocked. So the natural clot buster is a fibrin digesting enzyme called plasmin, and that's produced when the plasma protein plasminogen is activated. So it's sort of like a little self-destruction mechanism, and plasminogen is incorporated into a forming clot, and it stays there until there's signals that reach it. The presence of a clot in and around blood vessels causes the endothelial cells to secrete tissue plasminogen activator. And this activated factor and thrombin release during clotting also activates plasminogen. So as a result, most plasmin activity is confined to the clot, and circulating enzymes quickly destroy any plasmin that gets into the plasma at all. Fibrinolysis begins within 2 days and continues slowly over several days until the clot finally dissolves. Now again, I know this is an incredibly complicated process, and if you can believe it, I tried to pare down as much as I possibly could. But let's stick to the vascular spasm, the platelet plug formation, and coagulation. Finally, when the clot is done doing its job and everything is healed, we have fibrinolysis. You also may want to find some videos on YouTube or Google or something if you'd like to review this, but just remember, I'm not making you learn all the different clotting factors and the 2 pathways to prothrombin activator. I just kept it very, very general. Now, the only thing that's probably more important than having a clot, or at least equally important, is to inhibit clots when you shouldn't have them. So once a clotting cascade has begun, it continues until a clot forms. All right, and then we have to get rid of it very quickly. We get rid of those clotting factors really quickly. We inactivate all of the clotting factors, and if that wasn't enough, we do have things that prevent fibrin production when we shouldn't have it. And that includes antithrombin 3, a protein that's always present in your plasma. We have protein C, another protein that's produced in your liver. These inhibit the activity of the pathway of clotting factors coming together. We have heparin, which is a natural anticoagulant in those basophils we just talked about and in mast cell granules. It's also found in the surface of endothelial cells. Vitamins K and E also play a role in blocking fibrin production when you shouldn't have it. So these are all super important things, and there's actually a lot more than this. I just wanted to mention a few. One of the most interesting things about talking about blood are blood disorders, and I could talk all day about blood disorders, but I tried to stick to some of the more common ones. So a clot that develops and persists in an unbroken blood vessel, that is a clot that just stays in a blood vessel, it's called a thrombus. And if the thrombus is large enough, it can block circulation to the cells and the tissues can actually die. So an example of this would be a blockage in a coronary artery and an artery in your heart, and the heart muscle can actually die. That's what a heart attack is. We also have an embolus, which is basically when a blood clot or a thrombus breaks away and starts to float off in the bloodstream. So this can wedge just about anywhere and become an embolism and obstruct a vessel. And this could be in the heart, in the brain. You don't want it anywhere, right? But really bad. So we also have leukemia. Leukemias are cancers involving white blood cell, and it's basically a group of a whole type of cancers that involve the overproduction of abnormal white blood cells. Now, these abnormal white blood cells can't do what normal white blood cells can do, which is their job. And so as a result, we now have real problems with our immune system. The leukemias are named according to the rate at which they progress and the type of cell primarily involved. There are numerous different types of leukemias. So lymphocytic leukemia involves lymphocytes. Chronic leukemia is slowly advancing because it involves proliferation of later cell stages. The more serious acute forms of leukemia, acute means it's quickly advancing because it comes from stem cells. They affect children, unfortunately, and chronic leukemia is often more in elderly people. So without therapy, all leukemias are fatal, but the treatment and how quickly or severe they are just depends on the type of leukemia that it is. So the treatment can be anything from radiation to chemo to stem cell transplants and bone marrow transplants. It completely depends upon the leukemia, but often bone marrow or stem cell transplants are part of the treatment for it. Anemia, we've already mentioned when we talked about blood, but anemia are just real problems with erythrocyte production. And the problems can range from lack of raw materials like iron to failure of the red bone marrow to complete erythropoiesis. So anemia just means that the blood has a lower oxygen carrying capacity. Now this can be iron deficiency anemia, a really common one in the United States, which is basically you're just not getting enough iron and you can't make correct amounts of hemoglobin. The treatment is just to increase iron intake in the diet or through iron supplements. Other types of anemias, there's renal anemia, which is caused by lack of EPO, that hormone made by the kidneys that controls red blood cell production. This often accompanies renal disease or kidney failure or kidney damage. We have sickle cell anemia, which is genetic, where there's an abnormal type of hemoglobin, results in a change in just one of the 146 amino acids in the beta chain of the globin part of the hemoglobin molecule. And this causes our red blood cells to be shaped very strangely like half moons or a sliver of a moon, crescent shaped, and they have a really hard time carrying oxygen. These erythrocytes rupture easily and they tend to jam up blood vessels. There's a lot of different types of anemias, you guys. It could just be, pernicious anemia is an autoimmune disease that most often affects elderly people, where the immune system of these individuals destroys cells of their own stomach mucosa. And these cells normally produce, if you remember, intrinsic factor that has to be present for vitamin B12 to be absorbed. Without vitamin B12, erythrocytes grow but can't divide, and we end up with these really large pale cells that can't carry hemoglobin or oxygen the way that they need to. So that's just a few different types of anemia, but it's anything from lowered number of red blood cells, from blood loss, bone marrow failure, etc. Could be lower hemoglobin content, could be abnormal hemoglobin like in sickle cell, a lot of different things. So mononucleosis is a infection of B lymphocytes with the Epstein-Barr virus. So remember those bead lymphocytes, we talked about a type of white blood cell, and they're primarily responsible for making antibodies. Mono is often called the kissing disease. It's a highly contagious viral disease, the Epstein-Barr virus, and it really just causes people to have lots of infections that they can't fight. So a sore throat and a fever, and there really is no cure other than just resting and allowing the virus to run its course. Septicemia is when you have a bacterial infection in your blood. Now this is incredibly dangerous and can result in death, because if you have bacteria in your blood, you can have bacteria anywhere. Another name for septicemia is blood poisoning. It's just harmful levels of bacteria or their toxins in your blood. It is extremely dangerous. So I could talk for a long time about different blood disorders. That's just a couple. I hope that you've enjoyed the blood section. We're going to keep on keeping on with the cardiovascular system. Stay safe and study anatomy and physiology, everybody. Bye.